essentially, the glycolysis is broken down into two phases. The first phase is where you activate the glucose molecule. Once it's inside the cytoplasm, you activate it, you add a phosphate, and then you change it around a little bit in terms of its structure, and then you add another phosphate, and that's where the activation part of glycolysis is, from step 1 through step 3. Okay? The second half of glycolysis is where you start breaking the molecule down, the activated form, if you want, down to get energy in the form of either ATP or NADH. So glucose is a 6-carbon molecule, hexose, 6-carbon sugar. At the end of glycolysis, after 9 steps, you will get a 3-carbon keto acid called pyruvate. But you start with 6, so you have to balance that equation. So you get 2 pyruvates. 2 times 3 equals 6. Okay? You don't lose anything. There's no black hole anywhere. Okay? So you got to account for the carbons. Okay, so here's the big picture. We start with glucose. That's inside the cytoplasm. You activate it. Once you form this molecule, you actually trap it inside the cell. It can't go anywhere. Okay? Once you phosphorylate, you add a phosphate to glucose, it's no longer able to leave. Okay, so that's very important. Then you're going to go through an isomerization here, change glucose 6-phosphate to fructose 6-phosphate. Then you add another phosphate to form fructose 1,6-bisphosphate. These first 3 steps are, if you want, the activation of glucose to allow the rest of glycolysis to occur. The rest of the way, you're going to produce energy in the form of NADH. You're also going to produce energy in the form of ATP, not shown here. So you will have 2 types of energy molecule produced from glycolysis. This is the aerobic conditions, where you have enough oxygen, plenty of oxygen, and that's in most tissues. Anaerobic glycolysis, because of the lack of oxygen or lack of mitochondria in certain cells, the NADH ends up being used up to convert pyruvate to lactate. So under anaerobic conditions, you lose energy because the NADH is used up to convert pyruvate to lactate. I'll talk more about that in a minute. So here's the overall big picture of, you hear me okay? Okay, if I fade, let me know, because sometimes it's hard for me to know exactly how the sound is across there. Okay, so so here's the big picture, and that's really item number 3, if you want. We covered item 1 and 2. Where it takes place is in the cytoplasm and most tissues, all tissues. Item number 3 is energy. You use 2 ATPs initially in those preparative state, if you want, the energy investment phase. So you use up 2 ATPs. in the energy generating state or phase, you produce 4 ATPs. 4 minus 2, what you used up before, gives you 2 as net. You also produce 2 NADH molecules, high energy molecules. That's down here. And you can't forget the pyruvates. 2 pyruvates. So that's the overall yield from aerobic glycolysis. 2 NADH, 2 ATP, and 2 pyruvates. You can't hear? Better now? This mic keep changing positions on me. Okay. So this is the important net from aerobic glycolysis. 2 pyruvate, 2 ATP, 2 NADH. Again, the importance of pyruvate is pyruvate can be further oxidized later on by separate pathways, all the way to CO2. And those pathways are going to provide significant amount of energy. So, and this is an example of when you how different tissues metabolize glucose. For example, the brain will take a molecule. By the way, the brain's preferred energy source is sugar, is glucose. Okay? When you have a little cup of coffee in the morning or tea or whatever, if you have it with sugar, it's probably the sugar that's reviving you. Okay, rather than the caffeine, but they kind of work synergistically. Okay, so don't underestimate the value of sugar for your brain function. So the brain will take glucose and oxidize it first through glycolysis, then through pyruvate dehydrogenase, through the TCA cycle, all the way to the simplest possible molecule, which is CO2, with the purpose of getting every ounce of energy from glucose. Okay? Smart. The brain is smart. It wants to get as much energy out of glucose. The liver can do the same thing, but the liver has alternative pathways. It can, under specific conditions, go to lactate. It can actually use some intermediates along the way to make fat, to make fatty acids and triacylglycerol. Okay? But importantly, the liver, although this has nothing to do with glycolysis right now, the liver is uniquely able to make glucose, whereas glycolysis is to consume glucose. The liver can consume and make glucose. The only organ really to be able to do that, with the single exception of the kidney. So under specific conditions, the liver can make glucose and deliver it to the blood. Adipose tissue will break it down to acetyl-CoA and then can store much of that in the form of fat. Muscle, similar to the brain and liver, can go all the way to CO2 under anaerobic conditions, can produce lactate. This is unique here, erythrocytes. The last thing that erythrocytes can produce from glucose is lactate. They can't go any further. And the reason being is that erythrocytes or red blood cells don't have mitochondria. Mitochondria are very important in the conversion of pyruvate or oxidation of pyruvate all the way to CO2. All of that occurs in the mitochondria. And the conversion of high energy molecules like NADH and FADH2 into usable energy as ATP also occurs in the mitochondria. So the absence of mitochondria in red blood cells prevents them from using NADH. Therefore, they use that NADH to convert pyruvate to lactate. In essence, red blood cells are anaerobically performing glycolysis, and because of that, their output of energy is much less. They produce just 2 ATPs from glycolysis. The NADH is used up in the conversion of pyruvate to lactate. This is kind of the big picture. I'm not going to spend too much time on it, but I want you to see here's glucose, here's pyruvate. There's 9 steps. We're going to go through all of them here, but I want you to pay attention to a couple of things. And these things are in this yellow labels. And that really brings us to the item number 4, where is going to be very important. These here are rate-limiting enzymes. Step 1, there are 2 of them actually, hexokinase/glucokinase, 2 isozymes, PFK1, and pyruvate kinase. 3 regulated steps. Those steps you want to really pay attention to. Okay? And once we get to each one, I'll tell you what you want to pay attention to. Okay? The second group here, glyceraldehyde 3-phosphate dehydrogenase and phosphoglycerate kinase, these guys produce energy. So you want to know where the energy is produced. Okay? Always remember, your end point can be either pyruvate or lactate, depending on the aerobic conditions or anaerobic conditions. Pyruvate can be further degraded first through a step here, then through a TCA cycle, all the way to CO2. Okay? So that's the big picture, and now we can, again, 3 regulated steps, 2 pyruvates from glucose. Under anaerobic conditions, you produce lactate instead, and the NADH is used there. So here again, I'm going to go through. We've already covered that. I want to get started really on at least a couple of enzymes. So I just want to tell you that you have an energy investment phase, which means you have to use up energy in these steps to activate glucose. And that energy is used up in the form of ATP. You're going to add a phosphate first on glucose to the 6th position. Second, you're going to isomerize. You change this. This goes from a 6-member pyranoside to a 5-member furanoside, which is fructose. Fructose 6-phosphate. That's an isomerization. You haven't lost anything. You just reorganize the molecule. And then you use another ATP to add another phosphate at the 1 position of fructose. And this is really the activated molecule that will be used in the energy producing phase of glycolysis. Okay? So the energy investment phase uses 2 ATP. The energy payoff will produce 4 ATPs, 2 NADHs. So your net will be 2 ATPs and 2 NADHs plus 2 pyruvates. Okay. So here's the first step. First step of glycolysis. This is for simplicity. Again, you don't need to memorize structures or anything, but it's good to visualize what's happening. So here's glucose. That's carbon number 1 always. This is carbon number 6. So you get a phosphate from ATP. You add it to the 6th position, and that gives you glucose 6-phosphate. There are 2 distinct enzymes. The first one is called, you remember when we talked about isozymes? Well, they're really different proteins, have different tissue distribution, but they catalyze the same reaction. Okay? Hexokinase is present in most tissues. Hexokinase has different kinetic parameters. That's why we really spend so much time on Michaelis-Menten kinetics, Km and Vmax, because it's meaningful. It has a practical meaning for the cells, for the body tissues. Okay, hexokinase has a low Km, means high affinity for glucose, and low Vmax. So that means it saturates very quickly. But it can operate at very low glucose concentrations. And that's present in most tissues. One last important thing about hexokinase is that it's inhibited by the end product, glucose 6-phosphate. When this accumulates, it inhibits hexokinase. So all I said thus far are summarized here. Okay? So this is an important feature that you want to pay attention to. First one I forgot to mention, this is a one-way reaction, irreversible. You can't go back. Okay? All the rate-limiting enzymes will be irreversible. And that's the first one of them. Now, the second isozyme, glucokinase, is very important as well, but it has a more limited tissue distribution. And that tissue distribution is limited to the liver and pancreas. Okay? Liver and pancreas have glucokinase. Glucokinase parameters are, kinetic parameters are the opposite. It has high Km, means low affinity for glucose, and high Vmax, means it does not saturate as quickly. Okay? So the third thing is it's not inhibited by glucose 6-phosphate. So if you, since we're on this slide, this is really the key features you want to focus on. And that's going to be present on all the steps. Okay? So when in doubt, make sure you know these here. And that's, make sure you know that which one is where, and so on. This should look familiar. Does it look familiar? Okay. So, you know that the point of Michaelis-Menten equation and Michaelis-Menten graphs is to recognize one when you see one. Okay? So, the first thing you want to know about Michaelis-Menten plots is that the shape of the plot is hyperbolic. Here's hyperbolic and reaches a Vmax. This one too is hyperbolic and reaches a Vmax. It's just a much higher Vmax. Okay? So that's very important. to appreciate that what the shape of the Michaelis-Menten plot is. Let me start with hexokinase. We said hexokinase is in most tissues. Again, here's glucose concentration down here. That's enzyme activity. That's reaction velocity. Okay? Conversion of formation, if you want, of glucose 6-phosphate per minute, let's say. Okay? So look what happens with hexokinase. Let's say this one here is 1 millimolar. Once you get to 1 millimolar, it's almost completely saturated. It can't go any faster. So hexokinase with the low Vmax saturates quickly. That means it has a limited capacity to consume glucose. You can increase it all the way here to 10 millimolar. It's going as fast as it can. By contrast, I want you to look at glucokinase. Glucokinase has a high Km, low affinity. It's kind of sluggish here, going kind of slow. Look how low the initial velocity is, the velocity there. But once you start getting to, say, 4, 5, its activity increases linearly and continue to increase linearly for a long time, up to 10-15 millimolar of glucose. That's because it has a high Vmax. It does not saturate quickly. So why is that important? And how is that physiologically important? Well, it's important because look at this range there. That's the physiological blood glucose concentration. Around 4 to 5 millimolar. 5.5, 3.5 to 5.5. In that range, glucokinase can increase its activity as a function of increasing glucose concentration. So after a meal, let's say you were at 3.5. Before a meal, you were at 3.5. And after a meal, you went to 5.5 or 6. Glucokinase can handle that increase in glucose concentration and is able to pick up glucose from the bloodstream after a meal and metabolize it efficiently. Whereas hexokinase is already saturated, can't handle any more glucose. So the liver, because it contains glucokinase, is uniquely equipped to metabolize glucose from the bloodstream in this physiological range. That's the physiological range of glucose in the bloodstream. Okay, this is a very important concept. I hope you get it, but take some time, look at this one more time on your own and make sure you understand its significance. If you like this channel, please subscribe. Thank you.